The forest announces this one before it ever moves, not through sound, but through color, an interruption in the otherwise layered greens and browns of the Amazon floor. On a slick, moss-dark route where moisture beads into perfect spheres, something small holds its position with absolute confidence. It does not attempt to blend in, it does the opposite. Its body is saturated with color so intense it feels deliberate, almost excessive, as if the environment itself has chosen to highlight it. A poison dart frog, one of several species within the family Dendrobatidae, occupies this exposed surface without hesitation, its presence defined not by concealment, but by declaration. This is not carelessness. It is strategy refined to the point where visibility becomes a form of protection. In a system where countless organisms rely on camouflage to avoid detection, the poison dart frog adopts a different logic entirely. It advertises itself. The bright pigmentation, electric blues, deep blacks, radiant yellows, or saturated reds, functions as aposematic signaling, a biological warning that communicates toxicity to any predator capable of learning. These signals are not abstract. They are reinforced by experience, often harsh and immediate. A single contact, poorly judged, can create a lasting association between color and consequence. Yet the frog does not enforce this warning through aggression. It does not chase, does not bite, does not strike. Its defense is passive in motion, but active in chemistry. The skin, smooth and constantly moist, is infused with alkaloid compounds derived from its diet, primarily small arthropods such as ants, mites, and beetles that themselves carry defensive chemicals. Through a process of sequestration, the frog accumulates these compounds and stores them within specialized glands in the skin, effectively transforming what it consumes into a layered chemical barrier. This process ties the frog's toxicity directly to its environment. In captivity, where these dietary sources are absent, poison dart frogs often lose much of their potency. In the wild, however, the continuous intake of alkaloid-rich prey maintains a chemical profile that can be remarkably complex. Different species, and even populations within the same species, can exhibit variations in toxin composition depending on the availability of specific prey items. The frog becomes a living reflection of its ecological network, its defenses shaped by the diversity and distribution of the organisms it consumes. When contact occurs, the transfer is subtle. There is no injection, no visible wound, no immediate mechanical damage. The skin secretes minute amounts of toxin, enough to coat the surface without appearing wet or altered to the naked eye. Any organism that brushes against it, whether through a misstep, a predatory investigation, or simple proximity, may pick up these compounds on its own body. The entry point is often indirect. The toxin adheres to skin, to the sensitive lining of the mouth, or to the eyes if contact is made at that level. From there, it begins to interact with physiological systems in ways that are not always immediately apparent. Many of the alkaloids present in poison dart frogs target ion channels in nerve and muscle cells, particularly sodium channels that regulate electrical signaling. By binding to these channels, the toxins alter their normal function, preventing the controlled opening and closing that allows nerve impulses to propagate in a coordinated manner. The result is a disruption of communication within the body. Muscles may contract involuntarily or fail to contract at all. Sensory signals may become distorted, amplified, or suppressed. The organism is not simply experiencing pain or weakness. It is experiencing a breakdown in the system that allows it to interpret and respond to its environment. What defines this interaction within the context of the forest is not the immediacy of its effect, but the way it unfolds over time. The frog remains where it is, often unmoving, its role in the encounter complete the moment contact is made. The organism that has touched it, however, carries the interaction forward. The toxin, now present on or within its body, continues to influence physiological processes. Depending on the species and the amount of exposure, this can manifest as a gradual loss of coordination, irregular muscle activity, or, in severe cases, systemic disruption that compromises vital functions. In smaller predators, arthropods or amphibians that may attempt to consume the frog, the effect can be rapid enough to prevent successful predation. A brief grasp or bite is enough to initiate a cascade that discourages further contact. The predator releases the frog, often too late to avoid the initial transfer, and must then contend with the consequences. For larger animals, the interaction is often less immediately decisive, but still significant. A predator that mouths a frog and then releases it may not suffer catastrophic failure, but the experience is enough to reinforce avoidance. The cost is not always measured in survival or death, but in the disruption of normal function and the memory of that disruption. This is where the intelligence of the system becomes clear. The frog does not need to incapacitate every potential threat completely. It needs to create a reliable association between its appearance and an undesirable outcome. Over time, this association spreads through populations of predators, shaping behavior at a broader scale. Young or inexperienced animals may still make the mistake, but those that survive carry the lesson forward, reducing the likelihood of repeated encounters. The persistence of the toxin's effects plays a crucial role in this learning process. If the consequences of contact were brief and easily resolved, the cost of the mistake would be low and the signal would be weak. Instead, the alkaloids ensure that the body remains engaged in the aftermath. The disruption of nerve signaling does not simply switch off when the frog is no longer present. It continues until the compounds are metabolized, redistributed, or otherwise neutralized by the organism's physiology. During this time, the affected individual operates at reduced capacity, its interactions with the environment altered in ways that can have secondary consequences. The environment itself amplifies these effects. The Amazon is not forgiving of impairment. Movement through dense vegetation requires coordination. Foraging demands precision. Avoiding predators depends on rapid, accurate responses to stimuli that are often subtle and fleeting. An animal dealing with disrupted nerve signaling is at a disadvantage across all of these domains. It may move more slowly, misjudge distances, or fail to respond appropriately to threats. The initial contact with the frog becomes a pivot point, altering the trajectory of the animal's interactions with its surroundings. From the frog's perspective, this system allows for a remarkable degree of exposure. Unlike many small animals that rely on concealment to avoid detection, poison dart frogs can afford to occupy visible positions within the forest. They move during the day, when light penetrates the canopy and colors are most easily perceived. Their activity patterns align with the periods when visual predators are also active, reinforcing the effectiveness of their warning signals. This diurnal behavior sets them apart from many other amphibians, which tend to be nocturnal to avoid predation and desiccation. Thermoregulation in these frogs is also closely tied to their environment. As ectotherms, they rely on external sources of heat to regulate their body temperature. The combination of sunlight filtering through the canopy and the high humidity of the forest allows them to maintain activity without the risk of drying out. They select micro-habitats that balance exposure and moisture, often moving between shaded and partially lit areas to optimize their physiological state. This constant adjustment is subtle but essential, ensuring that their metabolic processes remain within functional limits. Reproduction adds another layer to their ecological role. Many species of poison dart frogs exhibit complex parental behaviors, including the transport of tadpoles to water-filled bromeliads or other small reservoirs. In some cases, parents provide unfertilized eggs as a food source for developing young. These behaviors extend the frog's influence beyond its own survival, shaping the micro-ecosystems in which its offspring develop. The same chemical defenses that protect the adult may also play a role in deterring predators from these vulnerable sites. Within this broader context, the poison dart frog represents a shift away from the more familiar narratives of predation based on force or speed. Its strategy is built on chemistry, visibility, and persistence. The moment of contact is almost incidental, often lacking the dramatic cues associated with a bite or sting. There is no sudden lunge, no visible wound. Yet, the consequences are real and, importantly, extended. The toxin continues to act, interacting with the body in ways that are not immediately resolved. This delayed unfolding aligns with a broader pattern seen across the forest, where many interactions are defined not by their initial impact, but by their after-effects. The frog's role within this pattern is distinctive because it operates entirely without the need for direct aggression. It does not need to escalate an encounter. It simply exists within a chemical framework that ensures any contact carries a cost. As the day progresses and the light shifts again, the frog remains in place or moves only slightly, its movements controlled and efficient. Around it, the forest continues its constant exchange of energy and information. Other organisms pass by, some recognizing the warning and adjusting their path, others unaware or inexperienced enough to come too close. The system does not eliminate risk. It redistributes it, placing the burden on those that fail to interpret the signals correctly. In this way, the poison dart frog embodies a form of ecological intelligence that relies on anticipation rather than reaction. It does not wait for a threat to fully materialize. It broadcasts its defense in advance, shaping behavior before contact occurs. And when that contact does happen, it ensures that the consequences extend beyond the moment, carried forward by the very systems that allow other organisms to function. The attack, if it can be called that, is quiet and often unseen, but its effects are sustained, unfolding within the body long after the frog has ceased to be part of the interaction. The forest floor in the Amazon rarely presents itself as empty. What appears at first glance to be a simple layer of damp leaves is, in reality, a dense, multi-layered system of shelter, decay, and concealment where movement is constant but rarely seen. Beneath the broad, decomposing leaves and the lattice of fine roots, moisture is trapped and held, creating a humid microclimate that favors organisms built for close contact with the ground. In this compressed world, vision becomes less important than vibration, chemical detection, and the ability to move through tight, resistant spaces. It is here, in this low and cluttered plain, that the giant centipede operates, not as a wandering predator, but as a precise, ground-level ambush specialist adapted to strike within centimeters of its target. A giant centipede of the genus Scolopendra does not announce itself through sound or display. Its body is segmented, flattened, and built to remain in constant contact with the substrate, allowing it to interpret subtle changes in pressure through dozens of legs that function as both locomotor tools and sensory extensions. Each step is information, each vibration passing through the leaf litter, whether caused by a struggling insect, a shifting amphibian, or a careless vertebrate footfall, feeds into a system that prioritizes speed and direction over deliberation. This is not an animal that stalks from a distance. It waits within reach, relying on the certainty that something will eventually pass too close. The head is the focal point of its predatory design. Just behind the antennae, which sweep continuously to sample chemical cues in the air and on surfaces, lie the forcipules, modified front legs transformed into venom-delivering appendages. They are not fangs in the traditional sense, but articulated limbs that curve downward and inward, capable of gripping and piercing simultaneously. When the centipede strikes, it does not simply bite. It hooks into its target, using the forcipules to anchor itself while venom is driven deep into tissue. This anchoring behavior is critical. In an environment where prey can thrash, jump, or roll, maintaining contact for even a fraction of a second longer can determine whether the venom is effectively delivered or wasted. The strike itself is almost always initiated from a low angle, often from partial concealment beneath leaf edges or within shallow depressions in the soil. The centipede's body compresses, storing elastic energy across its many segments, and then releases it in a rapid forward extension. There is no warning posture, no raised display. The motion is direct and efficient, minimizing exposure and maximizing contact time. The initial puncture is only the beginning. Almost immediately, the surrounding legs engage, wrapping partially around the target or pressing against the substrate to stabilize the centipede's body. This multi-point contact allows it to resist being dislodged while the venom begins to take effect. What makes this interaction particularly effective is not just the potency of the venom, but its role within a broader mechanical strategy. The centipede does not rely on venom alone to subdue prey. Instead, it combines chemical disruption with physical restraint, creating a layered attack that overwhelms multiple systems at once. The venom itself is a complex mixture of proteins, peptides, and enzymes designed to interfere with nerve signaling, muscle contraction, and cellular integrity. In smaller prey, this can result in rapid immobilization. In larger animals, the effect is more fragmented but no less disruptive. Localized paralysis, intense pain, and involuntary muscle contractions that compromise coordinated movement. For vertebrate prey such as small rodents, reptiles, or amphibians, the initial bite often triggers a violent escape response. The animal may lunge, twist, or attempt to bite back. The centipede's anchoring legs and low center of gravity are essential here, allowing it to remain attached long enough for the venom to begin altering the prey's behavior. Even if contact is broken quickly, the encounter does not end cleanly. The venom continues to act within the tissue, producing a cascade of effects that extend beyond the moment of the strike. Pain is one of the most immediate consequences. Unlike many predators that aim to silence or quickly incapacitate, the centipede's venom induces a sustained burning sensation that radiates outward from the bite site. This is not incidental. Pain, in this context, functions as a disruptive force. It interferes with normal movement patterns, distracts the animal from coordinated escape, and can lead to erratic behavior that increases vulnerability to further predation. Swelling follows, as inflammatory responses draw fluid into the affected area, altering tissue structure and limiting mobility. In small animals, this can be enough to tip the balance from escape to exhaustion. What is particularly striking is how the ecological role of the centipede extends beyond the immediate kill. In many cases, the centipede does not need to maintain prolonged contact with its prey. A brief, successful envenomation can create conditions that persist after the centipede has already withdrawn. The injured animal, now compromised by pain and impaired function, becomes easier for the centipede to track or for other predators to exploit. In this sense, the centipede's attack can initiate a delayed sequence of vulnerability, altering the dynamics of the surrounding micro-ecosystem. The forest floor amplifies this effect. Dense leaf litter restricts visibility and movement, forcing injured animals to navigate a complex terrain while already compromised. Moisture accelerates the spread of chemical cues, allowing predators to detect stress-related compounds released by struggling prey. Even the structure of the ground, soft, uneven, and layered, can exacerbate the difficulty of escape as footing becomes unreliable and obstacles multiply. Within this environment, a small wound can have disproportionately large consequences. The centipede itself remains largely unaffected by these secondary processes. After disengaging, it often retreats into cover, conserving energy and avoiding unnecessary risk. Its metabolism is adapted for periods of inactivity, punctuated by short bursts of intense activity. It does not need to chase extensively or engage in prolonged sangharsh. Instead, it relies on the predictability of movement within its territory. Given time, another opportunity will present itself. This approach reflects a broader pattern seen among many invertebrate predators in the Amazon: efficiency over spectacle, precision over endurance. The centipede's anatomy is not built for pursuit across open ground or for overpowering large resistant prey through brute force. It is built for close-range interaction, where speed, grip, and chemical disruption can be applied in a tightly controlled space. Its many legs are not redundant. They are a distributed system that allows for continuous contact and rapid adjustment, maintaining stability even when the substrate shifts or the prey struggles. From a physiological perspective, the centipede's venom delivery system also illustrates an important principle of evolutionary adaptation. Rather than maximizing toxicity alone, the system integrates delivery, retention, and effect. The forcipules ensure that venom is placed precisely where it can enter circulation quickly. The anchoring behavior extends the duration of contact just enough to improve efficacy. The venom composition itself balances immediate and delayed effects, ensuring that even incomplete envenomation can still produce meaningful disruption. In the broader ecological context, this strategy contributes to the regulation of small animal populations on the forest floor. By targeting a wide range of prey, arthropods, amphibians, reptiles, and occasionally small mammals, the centipede occupies a flexible predatory niche. It is neither a specialist nor a generalist in the strictest sense, but something in between, capable of adjusting its targets based on availability and opportunity. This adaptability is essential in an environment where conditions can shift rapidly with rainfall, temperature, and seasonal changes. There is also an element of asymmetry that defines these encounters. The physical size of the wound inflicted by a centipede is often minimal, especially when compared to the damage caused by larger predators. Yet the physiological and behavioral consequences can be extensive. This imbalance between visible injury and internal disruption is what gives the centipede its particular ecological impact. The mark it leaves behind is not a dramatic tear or fracture, but a point of entry for a process that unfolds over time. In the dim, humid layers of the Amazon floor, where visibility is limited and contact is inevitable, this kind of strategy proves effective. The centipede does not need to dominate the landscape or control large territories. It needs only to remain concealed, to interpret the signals that pass through its environment, and to act with precision when opportunity arrives. Its success is measured not in prolonged battles, but in brief, decisive interactions that alter the state of another organism long after the moment has passed. The result is a form of predation that feels less like a singular event and more like the beginning of a sequence. The strike is only the first step. What follows, pain, swelling, dysfunction, continues to shape the outcome, often out of sight, within the layered complexity of the forest floor. In this way, the giant centipede exemplifies a quieter but deeply effective form of survival, one that relies on the certainty that in the Amazon even the smallest contact can carry consequences that extend far beyond the point of origin. The ground here does not simply support life, it holds it in layers, storing heat, moisture, and movement within a compressed, living surface that never fully settles. Under a collapsed palm frond, where the soil remains dark and cool even under midday light, a burrow opens with no clear edge, only a subtle shift in texture where loose earth gives way to something deliberately shaped. This is not an abandoned cavity or a random hollow. It is a controlled environment, engineered over time by a large tarantula, often a species like Theraposa blondi, the Goliath bird-eater, whose presence is defined less by what it shows and more by what it prepares in advance. Inside, the air is stable. Temperature fluctuations are dampened by the insulating properties of soil, and humidity remains high, preventing desiccation in a body that relies on external moisture balance to function properly. The tarantula does not regulate its body temperature internally in the way mammals or birds do. Instead, it selects and modifies microhabitats that allow its physiological processes to operate within optimal ranges. This is thermoregulation through placement, not metabolism. The burrow becomes an extension of the spider's body, a buffer against the instability of the surface world. When it emerges, it does so without haste. The spider's movements are deliberate, each step placed with precision through a landscape that transmits information through contact. 8 legs spread outward, distributing weight and increasing sensitivity to vibration. The forest floor is not silent to the tarantula. It carries a constant flow of signals, footfalls, wing beats, the shifting of leaves under small bodies. These vibrations travel through the substrate, allowing the spider to interpret distance, direction, and intensity without relying on vision alone. Its eyes, clustered and relatively small, provide only limited detail. It is the ground that tells the story. Predation here is not based on pursuit, but on interception. The tarantula positions itself where movement is likely to pass, often near the edges of its burrow or along narrow corridors formed by roots and debris. When prey enters this zone, a wandering insect, a small amphibian, occasionally even a juvenile vertebrate, the spider responds with a rapid forward lunge. The strike is direct, the fangs penetrating quickly, delivering venom designed to immobilize and begin the process of external digestion. In many cases, the interaction ends here, the prey subdued and consumed with minimal resistance. But the tarantula's defensive strategy operates on a different axis entirely, one that does not rely on venom alone and often does not involve a bite at all. When threatened by a larger animal, something too large to subdue or consume, The spider shifts from predation to deterrence. The posture changes. The front legs may rise slightly, the body angling to present a broader profile. Yet the most effective component of this defense is less visible, carried not in the fangs, but across the abdomen. Covering the posterior section of the tarantula's body are thousands of specialized structures known as urticating hairs. These are not simple bristles. Each hair is a microscopic projectile, barbed along its length, designed to embed into the skin or mucous membranes of another organism. When the tarantula perceives a threat, it uses its hind legs to rapidly brush these hairs into the air, creating a fine, nearly invisible cloud that disperses outward from its body. The release is quick, almost incidental in appearance, but the effect is anything but immediate in the way a bite might be. The hairs settle onto exposed surfaces, skin, eyes, the lining of the nose or mouth, where they begin a process that unfolds over time. Unlike a venom that acts internally through the bloodstream or nervous system, urticating hairs operate at the interface between the organism and its environment. They do not need to penetrate deeply to be effective. Their barbed structure ensures that once they make contact, they resist removal, embedding themselves into tissue where they continue to interact with the body's defense mechanisms. The initial sensation may be subtle, easily dismissed in the chaos of movement through dense vegetation, but as the hairs lodge more securely, the body begins to respond. In the skin, this often manifests as irritation that intensifies gradually, a prickling sensation that evolves into a persistent itch or burning discomfort. The immune system recognizes the foreign material and initiates an inflammatory response, increasing blood flow to the area and recruiting cells designed to isolate and remove the irritant. This response, while protective, amplifies the sensation, turning a minor contact into a sustained source of distraction. In the eyes, the effect is far more severe. The cornea and surrounding tissues are highly sensitive, and the presence of barbed hairs can trigger intense pain, tearing, and involuntary closure of the eyelids. Vision becomes compromised, not because of direct damage in all cases, but because the body prioritizes protection over clarity. Blinking increases, tears attempt to flush the irritants away, and the affected individual may struggle to keep the eyes open long enough to navigate effectively. In the dense, uneven terrain of the forest floor, this loss of visual stability can have immediate consequences. What defines this interaction, however, is not the moment of exposure, but the persistence of its effects. The tarantula, having released its defensive cloud, does not need to remain engaged. It can retreat to its burrow or move away, conserving energy and avoiding further risk. The hairs, once deployed, continue to operate independently. They remain embedded, continuing to stimulate nerve endings and provoke immune responses long after the spider is gone. This creates a form of delayed suffering that aligns closely with the ecological pressures of the Amazon. An animal that has encountered a tarantula may not be immediately incapacitated, but its ability to function is compromised over an extended period. Grooming behaviors may increase as the animal attempts to remove the irritants, diverting time and attention away from foraging or vigilance. Movement may become cautious or erratic, particularly if vision is affected. Even minor reductions in efficiency can accumulate, especially in an environment where resources are contested and threats are constant. The effectiveness of this strategy lies in its economy. Producing urticating hairs requires energy, but once they are developed, their deployment is relatively low cost compared to the risks associated with physical confrontation. The spider does not need to match the size or strength of its opponent. It needs only to create a condition that discourages further interaction. Over time, this reinforces avoidance behaviors in other species. Animals learn to recognize the visual or chemical cues associated with tarantulas and adjust their movements accordingly, reducing the likelihood of repeated encounters. There is also an interplay between the tarantula's defensive mechanisms and the broader sensory environment of the forest. The air near the ground is often still, particularly under dense canopy cover, allowing the hairs to remain suspended briefly before settling. Moisture in the air can cause them to adhere more readily to surfaces, increasing the likelihood of contact. The complexity of the terrain means that an animal moving quickly is more likely to brush against surfaces where hairs have settled, extending the zone of effect beyond the immediate vicinity of the spider. From a physiological perspective, the interaction between urticating hairs and the body's immune system offers insight into how non-venomous defenses can achieve significant impact. The hairs themselves do not contain toxins in the way venom does. Instead, they rely on their physical structure to trigger a response. The barbs create micro-injuries as they embed, and the body's attempt to address these injuries leads to inflammation and irritation. This is a different pathway to discomfort, one that leverages the organism's own protective systems against it. In some cases, the effects can persist for days, particularly if the hairs are not fully removed. Secondary complications can arise if the affected area is repeatedly scratched or rubbed, leading to further tissue damage or the introduction of pathogens. In the humid conditions of the Amazon, where microbial life is abundant, even minor disruptions to the skin's integrity can create opportunities for infection. The original contact with the tarantula thus becomes the starting point for a chain of events that extends well beyond the initial moment. Within this framework, the tarantula's role in the ecosystem becomes more nuanced. It is not merely a predator of small animals, but a source of environmental pressure that shapes behavior through persistence. Its presence influences how other species move, where they choose to rest, and how they respond to certain cues. The cost of ignoring these signals is not immediate death in most cases, but a period of reduced capability that can be just as consequential. The forest floor, with its constant layering of interactions, amplifies these effects. An animal dealing with the aftermath of urticating hair exposure must navigate a landscape that does not accommodate weakness. Predators do not pause, and prey do not become easier to catch without consequence. The balance of the system depends on each participant maintaining a certain level of function. When that function is compromised, even temporarily, the risk profile shifts. The tarantula, meanwhile, returns to stillness. Its burrow remains a stable refuge, its body conserving energy, its sensory system attuned to the next vibration that signals opportunity or threat. It does not need to pursue or dominate. It has already altered the environment in a way that extends its influence beyond its immediate presence. The hairs it has released continue to act, embedded in tissue, provoking responses that outlast the encounter itself. In this way, the tarantula represents a different kind of interaction within the Amazon, one where the boundary between contact and consequence is stretched across time. The moment of exposure is brief, almost incidental, but the body carries it forward, reacting, adjusting, and enduring. The attack, if it can be called that, is not defined by a single action, but by the persistence of its effects. It becomes an ongoing story written not by the spider's continued presence, but by the body's inability to quickly resolve what has been introduced. The forest at ground level does not reward speed alone. It rewards tolerance. Tolerance for moisture, for decay, for the slow accumulation of biological processes that never fully reset. In a shallow depression where rainwater lingers just long enough to soften the soil without forming a permanent pool, something heavy shifts with deliberate calm. The shape resolves gradually. A broad, low body pressed close to the ground, skin textured like damp bark, eyes set high and outward, watching without urgency. The marine toad, often referred to as Rhinella marina, occupies this space not through stealth or aggression, but through a kind of ecological indifference. It does not need to chase. It does not need to hide particularly well. It simply remains, absorbing the environment into its physiology and waiting for the conditions that favor it. Its skin is not merely a covering. It is an interface, porous, chemically active, and constantly interacting with the surrounding humidity. Amphibians rely on this permeability for essential functions, including respiration and hydration. Water moves across the skin through osmotic gradients, and gases diffuse directly into the bloodstream without the need for complex lung ventilation alone. This dependence on external conditions makes the amphibian body both sensitive and adaptable, capable of exploiting microhabitats that fluctuate in temperature and moisture throughout the day. The marine toad positions itself where these gradients are stable, often at the edges of forest clearings or near temporary water sources, where insects gather and the air remains saturated. At first glance, nothing about the toad suggests escalation. Its movements are slow, its posture low and unthreatening, its coloration blending into the mottled tones of wet earth and leaf litter. Yet embedded within this unremarkable exterior is a defensive system that operates on a different principle than speed or force. Just behind the eyes, swelling outward like subtle ridges beneath the skin, are the parotoid glands, paired structures that store a concentrated mixture of bioactive compounds. These glands are not always active. They remain latent until the toad experiences pressure, stress, or direct contact that triggers their release. The mechanism is simple but effective. When compressed by a bite, a grasp, or even rough handling, the glands exude a milky secretion across the surface of the skin. This fluid spreads quickly, coating the immediate area and, depending on the interaction, transferring onto whatever has made contact. The toad does not need to aim or direct this process. It relies on proximity. Anything close enough to apply pressure becomes part of the exchange. The chemistry of this secretion is complex, composed primarily of bufotoxins, a group of compounds that include bufadienolidase and biogenic amines. These substances are not designed to tear tissue or cause immediate structural damage. Instead, they interfere with physiological processes at a systemic level. Bufadienolides, for example, affect the sodium-potassium pumps in cell membranes, mechanisms essential for maintaining the electrochemical gradients that allow cells, particularly in the heart and nervous system, to function properly. When these pumps are disrupted, the balance of ions shifts, leading to altered electrical activity across tissues. In a controlled medical context, compounds that influence these pumps can be used therapeutically in precise doses. In the uncontrolled context of a defensive secretion, the same mechanism becomes a source of instability. The body receiving the toxin does not regulate the exposure. It absorbs what is available, and the effects begin to propagate from the point of contact outward. Unlike a bite or sting, where the entry point is localized and often visible, exposure to the marine toad's toxins can occur across multiple surfaces simultaneously. The eyes, with their thin protective layers and constant moisture, are particularly vulnerable. A small amount of secretion transferred to the cornea can trigger immediate irritation, followed by intense discomfort as the compounds interact with sensitive nerve endings. Vision becomes blurred, not only from the chemical interaction, but from the body's response: tearing, blinking, and involuntary closure of the eyelids as protective reflexes activate. The mouth and mucous membranes present another pathway. Animals that attempt to bite or consume the toad often receive the highest dose, as the secretion is drawn directly into the oral cavity. Here, absorption is rapid. The lining of the mouth and throat allows compounds to enter the bloodstream more efficiently than through intact skin. The result can be a cascade of symptoms that extend beyond localized irritation: excessive salivation, nausea-like responses in species capable of such reactions, and disruptions in normal muscle coordination. Even without ingestion, the toxin can enter through small cuts or abrasions in the skin. In the Amazon, where surfaces are rarely clean and minor injuries are common, this pathway is not insignificant. The compounds do not need a large opening. The combination of moisture and repeated contact increases the likelihood that they will find a route into the body. Once there, they begin to interact with cellular processes in ways that are not immediately resolved. What defines the marine toad's role in this context is not the intensity of the initial exposure, but the persistence of its effects. The toad itself remains passive after the release. It does not pursue, does not escalate, does not maintain contact. It simply withdraws into stillness, its defensive act complete. Yet the organism that encountered it continues to process the consequences. The toxins, now present within tissues or on sensitive surfaces, continue to disrupt normal function. In predators, this often manifests as a delayed realization of error. A snake that has seized a toad may release it quickly, sensing something is wrong, but the exposure has already occurred. The toxin, transferred during the brief contact, begins to act along the lining of the mouth and through any absorbed pathways. Muscle coordination may falter. The precise timing required for effective movement, striking, constricting, or even simple locomotion becomes less reliable. The predator is not immediately incapacitated, but it is altered, its efficiency reduced at a critical moment. For mammals, particularly those that investigate with their mouths or noses, the experience can be equally disruptive. The nose, rich in sensory receptors and constantly moist, provides an efficient surface for absorption. Contact with the toxin can lead to irritation that spreads beyond the initial point, affecting breathing patterns and sensory perception. The animal may shake its head, rub its face against the ground or vegetation, attempting to remove the source, but in doing so may spread the toxin further across its own body. The unfairness of the interaction lies in this asymmetry. The toad does not need to dominate or even actively resist in a visible way. Its defense is built into its surface, triggered by contact, and sustained by the chemistry it releases. The attacker, having already disengaged, remains within the sphere of effect, dealing with a process that continues independently of the toad's presence. Within the ecosystem, this strategy has significant implications. The marine toad occupies a position that is not defined by speed or predatory dominance, but by resilience and deterrence. Its slow movements and exposed posture would make it vulnerable in a system driven purely by physical interaction. Instead, its chemical defense shifts the balance, allowing it to exist in open or semi-exposed environments where other animals must remain cautious. This, in turn, influences the behavior of surrounding species. Predators that have experienced the effects of the toxin may avoid similar shapes or scents in the future, altering their foraging patterns. The toad becomes a moving boundary, a living signal that carries with it the memory of previous encounters. Even without direct interaction, its presence can shape movement through the landscape, creating zones of reduced predation pressure that other organisms may exploit. The toad's own physiology supports this role. Its ability to tolerate a wide range of environmental conditions allows it to remain active in areas where water levels fluctuate and temperatures shift. It can conserve moisture through behavioral adjustments, retreating into burrows or shaded areas during dry periods and emerging when conditions improve. This flexibility ensures that it remains a consistent presence within its chosen habitat, reinforcing the effectiveness of its defensive strategy over time. There is also a broader pattern at play in the way chemical defenses operate in the Amazon. Unlike mechanical defenses, which often rely on immediate and visible interactions, chemical systems can extend their influence beyond the moment of contact. They operate across time and space, carried by moisture, absorbed through surfaces, and processed by the bodies they affect. The marine toad exemplifies this approach. Its toxins do not need to be delivered with precision. They need only to be present, ready to transfer when the conditions align. In the layered environment of the forest floor, where contact is inevitable and visibility is limited, this kind of defense proves particularly effective. The toad does not need to anticipate every potential threat. It prepares for contact as a certainty and ensures that when it occurs, the response will continue beyond the immediate interaction. The result is a form of persistence that mirrors the broader dynamics of the ecosystem itself. Processes that begin in one moment and unfold over many, shaping outcomes in ways that are not always immediately visible. As the light shifts and the moisture in the air thickens with the approach of evening, the toad remains in place, its body absorbing and releasing water in slow equilibrium with its surroundings. Around it, insects move, small vertebrates pass, and the constant exchange of energy continues. The toad does not need to assert itself within this system. It has already established its terms. Contact is the trigger, chemistry is the method, and time is the medium through which its influence persists. In a landscape where survival often depends on immediate reactions, the marine toad introduces a different equation, one where the consequences of a single moment can extend far beyond the point at which it occurred. The air beneath the Amazon canopy carries more than humidity. It carries signals, layered, overlapping, and persistent, moving through heat gradients, along bark surfaces, and across the invisible corridors that insects use to navigate a world defined less by sight than by chemistry. On the trunk of a mid-story tree, where buttress roots fold into the soil and moisture collects in thin films, a colony of bullet ants moves with deliberate cohesion. Their bodies are large by ant standards, each individual armored in a dark, matte exoskeleton that reflects very little light, making them easy to overlook against the bark. Yet nothing about their behavior is passive. Every movement is part of a distributed sensory system that extends across the colony, linking individuals into a single, responsive network tuned to disturbance. Bullet ants, members of the genus Paraponera, occupy a niche that combines arboreal and terrestrial pathways. Their nests are often located at the base of trees, but their foraging routes extend upward along trunks and outward across the forest floor. This vertical integration allows them to exploit multiple layers of the ecosystem, accessing nectar, small arthropods, and plant exudates, while maintaining a central defensive hub. The structure of the colony reflects this dual role. Workers are not specialized into rigid castes as seen in some other ant species. Instead, they maintain a flexible behavioral repertoire, capable of switching between foraging and defense depending on the signals they receive. Those signals are chemical first, mechanical second. Antennae sweep constantly, sampling pheromone trails laid by nest mates, detecting minute changes in the concentration gradients that define safe routes and active zones. Vibrations traveling through the bark or soil are interpreted through the legs, allowing the ants to detect approaching animals before they come into view. When a disturbance crosses a certain threshold, pressure, heat, or chemical intrusion, the colony shifts rapidly. Individuals that were moments ago engaged in routine foraging redirect toward the source, guided by alarm pheromones that spread outward in expanding waves. The response is not chaotic, it is structured escalation. Initial contact often involves a small number of workers investigating the disturbance, their bodies angled forward, antennae extended. If the intrusion persists or intensifies, more ants converge, forming a dense cluster that moves with surprising speed. Their goal is not to chase, but to make contact, to establish a point of interaction where their primary defensive tool can be applied. That tool is not their mandibles, though those are capable of gripping. It is the sting, a modified ovipositor connected to a venom gland that produces one of the most intensely painful chemical defenses known in the insect world. The sting of a bullet ant is delivered with precision rather than repetition. Unlike bees, which leave their stinger embedded and die after a single use, bullet ants can sting multiple times. The stinger itself is smooth, allowing it to penetrate and withdraw without becoming lodged in the skin. This mechanical design supports a strategy focused on mobility. The ant does not need to remain attached for long. It needs only to make contact, deliver venom, and disengage, leaving the biochemical process to unfold independently. At the moment of penetration, the venom enters the tissue and begins interacting with the nervous system almost immediately. The primary active component, a peptide known as ponerotoxin, targets voltage-gated sodium channels in nerve cells. These channels are critical for the generation and propagation of electrical signals along neurons. By interfering with their normal function, the toxin disrupts the controlled flow of ions that underlies nerve signaling. The result is a cascade of uncontrolled or prolonged depolarizations. Essentially, the nerves begin firing in a way that is no longer regulated by the body's usual mechanisms. This disruption manifests as pain, but not the brief, sharp pain associated with a mechanical injury. It is sustained, radiating, and often described in terms that reflect its persistence rather than its intensity alone. The sensation can spread beyond the immediate site of the sting, following nerve pathways and creating a field of discomfort that extends across a limb or region of the body. This is not incidental. From an evolutionary perspective, a defense that continues to operate after the ant has disengaged increases the likelihood that the intruder will withdraw and avoid future contact with the colony. What distinguishes the bullet ant's sting within the broader context of Amazonian defenses is the duration of its effect. Many venomous interactions in the forest are designed to incapacitate quickly, either to subdue prey or to deter predators through immediate discomfort. The bullet ant operates on a different timeline. The initial sting is only the beginning of a prolonged physiological event that can last for hours. During this time, the affected individual may experience tremors, localized weakness, and a reduction in fine motor control. Muscles near the sting site can become difficult to coordinate, not because they are physically damaged, but because the signals controlling them are being distorted. In vertebrates, including humans, this can create a paradoxical situation. The source of the disturbance, the ant, may be long gone, yet the body continues to react as if the threat is still present. The nervous system, caught in a loop of altered signaling, maintains a state of heightened activity that the organism cannot easily shut down. This is where the ecological function of the sting becomes particularly effective. It decouples the defensive act from the presence of the defender. The colony does not need to maintain a prolonged engagement. It can deliver its deterrent and return to normal activity while the intruder deals with the consequences. Within the forest, this has broader implications for how animals interact with their environment. Many species learn to recognize and avoid bullet ant colonies not through repeated direct encounters, but through the memory of a single, prolonged experience. The cost of contact is not measured in immediate injury, but in the extended disruption of normal behavior. An animal that has been stung may become less effective at foraging, slower to react to predators, or more cautious in its movements, all of which can influence its survival in subtle ways. The placement of colonies at the base of trees further amplifies this effect. These locations are natural convergence points for a wide range of animals. Mammals pass by while navigating the forest floor, reptiles use the roots as cover, and amphibians seek the moisture retained in the soil. Even birds may come into contact with these zones while foraging at low heights. By situating themselves in these high-traffic areas, bullet ants maximize the likelihood of encounters while minimizing the need to pursue or actively hunt large targets. There is also an interplay between the ants and the plants they inhabit. Some tree species appear to tolerate or even benefit from the presence of aggressive ant colonies at their base. The ants, in defending their territory, may deter herbivores or other organisms that would otherwise damage the plant. This is not a strict mutualism in all cases, but it reflects a broader pattern in the Amazon where defensive behaviors of one species can indirectly support the survival of another. The bullet ant, while primarily concerned with its own colony, becomes part of a larger network of interactions that shape the structure of the forest. From a physiological standpoint, the persistence of the sting's effects highlights an important aspect of venom evolution: the balance between potency and duration. A venom that acts quickly but fades rapidly may be sufficient for certain defensive scenarios, particularly when the goal is to create a brief window for escape. In contrast, a venom that induces long-lasting effects can alter the behavior of an intruder well beyond the immediate encounter. In the case of the bullet ant, the latter strategy appears to be favored. The colony invests in a chemical defense that continues to function after the initial delivery, extending its influence without requiring additional energy expenditure. This approach also reduces risk to the ants themselves. Prolonged physical engagement with a large animal would expose them to injury or death. By relying on a sting that can be delivered quickly and effectively, they minimize the time spent in direct contact. The ability to sting multiple times further enhances this strategy, allowing individuals to contribute to the defense without sacrificing themselves in the process. In the layered complexity of the Amazon, where countless interactions occur simultaneously and often unseen, the bullet ant's method of defense stands out, not for its spectacle, but for its persistence. The encounter is brief, almost incidental, a momentary contact at the base of a tree, a step placed too close to a nest entrance, a hand brushing against bark. Yet the consequences unfold over a much longer time scale, shaping behavior, memory, and movement in ways that extend far beyond the initial event. The forest does not pause for this process. While the affected animal contends with the ongoing effects of the venom, the surrounding ecosystem continues its constant motion. Other predators hunt, prey animals forage, and environmental conditions shift with the passing of clouds and the movement of air. Within this dynamic system, the bullet ant's strategy ensures that its influence persists even in its absence. It does not need to dominate the landscape or engage in repeated conflict. It needs only to deliver a single, effective signal, one that the body cannot ignore and cannot quickly resolve. In this way, the bullet ant embodies a form of ecological pressure that operates through time rather than force. The sting is not the end of the interaction, but the beginning of a prolonged state in which the organism must adapt, endure, and eventually recover. The imbalance between the simplicity of the act and the complexity of its aftermath is what gives it weight. A small insect, acting within the narrow confines of its territory, can impose a physiological burden that reshapes the behavior of much larger animals. In the Amazon, where survival often depends on the ability to respond quickly and efficiently to immediate threats, this kind of lingering disruption represents a different kind of challenge, one that cannot be escaped simply by moving away. The forest does not end with size. It narrows, compresses, and refines itself until the largest forces give way to the smallest decisions, where a fraction of a second, a millimeter of distance, or a single unnoticed surface becomes the entire outcome. On the underside of a broad leaf, where condensation gathers in trembling droplets and light filters through in diffuse green, something waits without declaring itself. It does not need color to warn or bulk to intimidate. It relies on absence, absence of movement, absence of contrast, absence of anything that might separate it from the surface it occupies. An assassin bug, a member of the family Reduvidae, holds position along a vein of the leaf, its body aligned so precisely with the structure that it becomes part of the plant rather than an organism resting upon it. This is camouflage at its most disciplined. The insect's form is angular but subdued, its coloration muted into browns, greens, or mottled patterns that echo the textures of bark, leaf, or lichen. Some species go further, collecting dust, plant fibers, or even the remains of their prey and adhering them to their bodies, constructing a layered disguise that disrupts any recognizable outline. The result is not invisibility in the absolute sense, but a form of perceptual silence. Movement passes over it without recognition because there is no signal to trigger attention. The assassin bug does not patrol or search in the way larger predators might. It positions itself within a corridor of inevitability. Leaves, stems, and narrow pathways in the forest act as funnels for movement. Insects travel these routes repeatedly, guided by scent trails, light gradients, or the structural logic of the plant itself. The assassin bug selects a point along one of these routes and commits to stillness. Time becomes an ally. The longer it remains undetected, the more likely it is that something will cross the threshold it has defined. Its sensory world is tuned to proximity. Antennae extend forward, sampling the air and surfaces for chemical cues, while fine hairs along the body respond to minute shifts in airflow and vibration. Unlike predators that rely on long-range detection, the assassin bug operates within a narrow window where reaction speed matters more than anticipation. When a potential target enters this space, a passing insect, a resting arthropod, or even the exposed skin of a much larger animal, the transition from stillness to action is immediate and precise. The strike is not dramatic. There is no sweeping motion, no visible buildup of force. The insect's head tilts, and from beneath it extends the rostrum, a specialized, needle-like mouthpart composed of interlocking segments. This structure is both rigid and flexible, capable of piercing through cuticle or skin with controlled pressure. When it makes contact, it does not tear or slice. It penetrates cleanly, guided by the alignment of its segments and the force generated by the insect's compact musculature. At the moment of penetration, the assassin bug delivers its defining tool, saliva, that functions as both venom and digestive agent. This fluid is not a single compound, but a mixture of enzymes and toxins designed to break down tissue and disrupt cellular integrity. In prey, the effect is immediate and often decisive. The saliva begins to liquefy internal structures, allowing the bug to consume its meal externally by drawing in the resulting fluid. The process is efficient, minimizing the need for prolonged struggle. But the role of this saliva extends beyond feeding. In defensive interactions, when the target is too large to consume or the contact is accidental, the same compounds produce a very different outcome. The penetration may be brief, almost unnoticed in the moment, especially if it occurs through a thin layer of skin or at a point with fewer nerve endings. The insect does not need to remain attached, like the systems seen earlier in the forest. Its effectiveness lies in what continues after it disengages. The saliva, once introduced into the tissue, begins to act on a microscopic scale. Enzymes break down cell membranes, disrupting the structural integrity of the affected area. Toxins interfere with local nerve signaling, creating a sensation that evolves rather than peaks. What begins as a small, localized irritation can intensify into a deeper, more persistent discomfort, often described as disproportionate to the size of the wound. The body responds with inflammation, increasing blood flow to the site and recruiting immune cells to contain and repair the damage. This is where the assassin bug strategy reveals its full impact. The wound it creates is not defined by its entry point, but by the process it initiates. The tissue does not simply close and recover. It reacts, sometimes overreacts, to the presence of foreign compounds that continue to alter its state. Swelling may develop, accompanied by a spreading sensitivity that makes the area difficult to ignore. In some cases, the breakdown of tissue can lead to small lesions or areas of necrosis, particularly if the enzymes have had time to act before being neutralized. For small animals, this can be enough to compromise function. An insect that has been pierced may lose the ability to coordinate its movements, not because of a large-scale injury, but because critical tissues have been disrupted at a localized level. For larger organisms, including mammals, the effect is more often one of persistent irritation and pain, a reminder that continues to demand attention long after the insect has moved on. The assassin bug does not remain to observe this outcome. It retracts its rostrum, shifts its position if necessary, and returns to stillness. Its energy expenditure is minimal, its risk controlled. The environment absorbs the event almost immediately. Leaves continue to sway, moisture continues to condense and evaporate, and the countless other interactions of the forest proceed without interruption. Yet, within the body that was pierced, the process continues. This delayed unfolding is particularly effective in an ecosystem where attention is a limited resource. An animal dealing with a persistent irritation or localized pain must allocate time and energy to address it. Grooming behaviors may increase as the organism attempts to clean or soothe the affected area. Movement patterns may change, either to avoid further contact or to compensate for discomfort. These adjustments, while small in isolation, can accumulate, altering the animal's efficiency in ways that ripple through its interactions with the environment. The assassin bug's ecological role is not confined to these defensive encounters. As a predator, it occupies a position that bridges multiple trophic levels. It preys on a wide range of arthropods, including species that themselves are predators or vectors of disease. By regulating these populations, it contributes to the balance of the micro-ecosystems it inhabits. Some species have even developed specialized relationships targeting particular types of prey with remarkable consistency, suggesting a level of ecological tuning that goes beyond opportunistic feeding. There is also variation within the family that reflects different adaptations to the same fundamental strategy. Some assassin bugs are strictly arboreal, operating along leaves and branches, while others inhabit the forest floor or even the edges of human structures. A subset, known as kissing bugs, has evolved to feed on vertebrate blood, introducing an additional layer of complexity through their role as vectors for pathogens. While these species are more commonly associated with other regions, they illustrate the versatility of the basic design: a piercing mouthpart, a potent saliva, and a reliance on proximity and timing. From a physiological standpoint, the efficiency of the assassin bug's feeding and defense mechanisms highlights the advantage of combining mechanical precision with chemical action. The rostrum ensures that the saliva is delivered exactly where it will be most effective, reducing waste and maximizing impact. The composition of the saliva, in turn, allows the insect to extract nutrients or deter threats without the need for sustained physical engagement. This integration of form and function is a recurring theme in the Amazon, where energy efficiency often determines success. The closing truth of this system is not dramatic, but it is consistent. The scale of an organism does not define the scale of its impact. A large predator may dominate through force, creating visible and immediate consequences. The assassin bug operates on a different principle. It creates a small point of entry and allows chemistry and physiology to carry the interaction forward. The result is a wound that continues to evolve, shaped as much by the body's response as by the initial action. In the dense, layered environment of the forest, where countless interactions overlap and intersect, this kind of precision has a quiet power. It does not rely on spectacle or repetition. It relies on the certainty that contact will occur and that even the smallest breach can become a pathway for change. The organism that caused it may already be gone, indistinguishable once more from the surface it inhabits, but the effect remains, unfolding within the boundaries of another body. As the light fades and the humidity thickens, the assassin bug maintains its position or shifts to another point along the same invisible pathways. It does not need to adapt its strategy to changing conditions. It is already aligned with them. Movement will continue, contact will happen, and when it does, the outcome will not be determined by size or visibility, but by the precision of a single action and the persistence of what it leaves behind.